Dear students, welcome to this lecture on edible films and coatings from plant and animal origin proteins. Shiny apples, crispy french fries and fresh nuts are the preferred foods for comparing to the least quality foods. For maintaining this acceptable nature, we can find food coatings as solution. Food coatings can be categorized as visible and invisible. These food coatings are maintaining the freshness and taste of the food products. Food coatings should be edible one. Some of the food coatings are having taste and some are not having any taste. Food coatings are having various applications in food such as to limit moisture transfer and oil transfer, prevent oxidation and stabilize the food during freezing and thawing. Wax on various types of fruits is one of the food coatings and used for preventing moisture loss and creating shiny fruit surface for highest consumer acceptability. Why do we need edible films? We are getting most of the foods directly from nature and many of the foods should be eaten immediately after harvesting. Food product is reaching the table of the consumer in a considerable time. If there is no special protection, damage can occur within hours or days during handling processes and products start to lose its sensory characteristics and nutritional value. Various advancements such as increased transportation and distribution facilities, storage methods, larger supermarkets and warehouse stores are providing longer shelf life to the food products. Objective of this lecture is that to understand the definition of edible films and coatings and its application. Identifying different types of plant and animal protein based edible films and coatings. Let us first look at the definition of edible coating and films. Any type of material used for enrobing, that is coating or wrapping various food to extend shelf life of the product that may be eaten together with food with or without further removal is considered as an edible film or coating. In detail we can say edible films give replacement or fortification of natural layers of food material to prevent moisture losses while selectively allowing for controlled exchange of important gases such as oxygen, carbon dioxide and ethylene, which are having important role in the respiration processes. A film or coating is used to prevent loss of other important components and surface sterility also. Thickness of edible coatings and film is less than 0.3 mm generally. And we must to know how to prepare this edible coating and film. Coating can be accomplished in various ways. In first method, the product can be dipped in film ingredients containing solution or it can be brushed or sprayed with solution. So the edible film will be directly deposited on food surface. In second method, stand-alone film can be created from solution or thermoformation can create covering for the food surface. Now let us discuss about components of edible films. Food components such as carbohydrates, proteins and lipids can be used for preparation of edible films. Lipids are used to reduce water transmission. Polysaccharides are used to control oxidation and other gas transmission, and protein films are used for giving mechanical stability. These ingredients can be used as either individual components or mixed composite blends to create edible films. They do not adversely modify food flavor. In preparing films for the food products such as fresh fruit and vegetables, main determination is to confirm that the developed films should provide physical and chemical properties to permit the gas and liquid transfer at the same rate in their fresh stage. Edible films should not contain any toxic, allergic and non-digestible components, and it should provide structural stability and prevent mechanical damage to the product during handling process. In different components of edible films and coatings, we are focusing about protein in this session. Proteins are the macromolecules and commonly used edible film and coating materials. They are having specific amino acid sequences and molecular structures. Proteins are having secondary, tertiary and quaternary structures of proteins, and these structures can be easily altered by heat denaturation, mechanical treatment, pressure, acids, alkalis, salts, metal ions, chemical hydrolysis, enzymatic treatment and irradiation. Comparing to other coating materials, proteins are having distinctive characteristics such as electrostatic charges, conformational denaturation and amphiphilic nature. Conformation of protein is affected by the important factors such as charge density and hydrophilic hydrophobic balance. These factors can eventually control the physical and mechanical properties of prepared films and coatings. Which protein sources are used for preparing edible films and coatings? Protein edible films and coating materials are derived from many different animal and plant sources such as milk, egg, animal tissues, grain and oil seeds. First, we will see different types of plant protein materials used for preparing edible films and coatings. Corn, soybean, wheat, cottonseed and other crops are used to extract protein to form edible films and coating materials. Different types of plant protein based materials are widely used such as zinc film and coatings, soy protein films, wheat gluten films, cottonseed protein films, other protein films. Let us discuss few points about the different types of plant protein based edible films and coatings one by one. First one is zinc film and coatings. In this, we should know what is zinc. Zinc embraces a group of prolamines which are found in corn endosperm. Commercially used zinc is obtained from the corn bed milling industry as a byproduct. After cleaning, corn is steeped in water containing sulfur dioxide to soften the grains. Coarse milling is carried out to separate the hulls and germ from the endosperm, and it is continued to get fine slurry. After that, the endosperm slurry is to be centrifugally separated to get a protein rich mass or corn gluten meal. Zinc can be extracted by using aqueous alcohol from the separated protein rich mass and dried to a granular powder. Now we will look about zinc coatings. Zinc is one of a commercially used edible coating like collagen and gelatin. Zinc coatings are used for confectionery foods and in pharmaceutical tablets. Zinc coatings are prepared in 3 steps. Zinc powder is dissolved in warm aqueous ethyl alcohol or isopropanol. Approved plasticizers such as propylene glycol or glycerin are added with the solution to enhance coating flexibility. The products to be coated are dipped into or sprayed with or brushed with the zinc plasticizer solutions. Antioxidants such as BHT or BHA are incorporated to prevent lipid oxidation, and vegetable oils to enhance shiny appearance. Zinc coatings are widely used in pharmaceutical coating and confectionery glazing. Next is zinc films. Zinc films are easily cast from alcohol solutions. Zinc is dissolved in warm aqueous ethanol or isopropanol with added plasticizers. The solution is cooled to 40 to 50 degrees Celsius, allowing bubbling to stop prior to casting, and is then poured over a glass plate where a film is formed as the alcohol evaporates from the surface. The dried film is finally peeled off from the plate. Plasticizers are incorporated to reduce film brittleness. They reduce the intermolecular forces between polymer chains, thus increasing flexibility and extendability. The most commonly used plasticizer for gene films is glycerol. The second type of plant protein based material is soy protein films. Based on its protein content, soy protein used in the food industry is classified as soy flour, concentrate or isolate. Defatted soy flour is obtained by grinding defatted soy flakes, and it contains 50 to 59% of protein. Soy protein concentrate is obtained by aqueous liquid extraction or acid leaching processes, and it contains 65 to 70% of protein. Soy protein isolate is obtained by aqueous or mild alkali extraction followed by isoelectric, and it contains more than 90% of protein. Soy protein is a feasible and renewable resource for producing edible and environmentally friendly biodegradable films. Soy protein films are prepared from soy protein isolate by drying thin layers of cast film forming solutions. The drying temperature and relative humidity that determine the drying rate of cast solution can also affect the film structure and properties. Rapid drying at 95 degrees Celsius and 30% relative humidity yields films that are stronger, thinner, stiffer and less extendable than films dried more slowly at 21 degrees Celsius and at 50% relative humidity. Soy protein films need the addition of plasticizers such as glycerol and sorbitol for texture and flexibility. By using soy protein isolate, plasticizer and water as the main formulation ingredients, extrusion of soy protein formulations can be done. This extrusion process is also done to get films by using the blends of soy protein isolate, polyethylene oxide and low density polyethylene. In multi-layer packaging, soy protein films are acting as an adhesion barrier providing layer. Soy protein isolate coating controls lipid oxidation and control surface moisture loss in pre-cooked meat products. Soy protein isolate coatings with incorporated antioxidants and flavoring agents enhance the overall quality characteristics of food products. Protective soy protein isolate coatings are providing high permeability to water vapor for the products such as high moisture, low sugar cakes and meat pies. Other applications of soy protein isolate films are micro-encapsulating agents of flavors and pharmaceuticals, or in coatings of fruits, vegetables and cheese. Let us discuss about wheat gluten films. Wheat gluten proteins are having high molecular weight, markedly apolar character, complexity and diversity of their fractions. These features are responsible for selective gas barrier properties and rubber-like mechanical properties of a flame. Wheat gluten-based materials are transparent, mechanically strong, homogeneous and relatively water resistant. They are edible, biocompatible and biodegradable with added food grade additives. Wheat gluten is an industrial byproduct of wheat starch production via wet milling. Wheat gluten is the cohesive and elastic mass, and it is obtained from the process of washing away the starch from wheat flour dough. Now we will see how wheat gluten films are prepared. Wheat gluten films can be acquired by casting from solution or by extrusion method. In these two methods, extrusion is more cost effective. Heating of wheat gluten solutions to nearer to boiling temperature creates surface skin, and this can be utilized as a flame. Sorbitol, mannitol, diglycerol, polypropylene glycol, triethylene glycol, maltitol, polyvinyl alcohol and polyethylene glycol are used as plasticizers for wheat gluten films. But glycerol and ethanolamine are desired. Wheat gluten based materials are having wide applications such as coating of foodstuffs, seeds and pills, and for making cosmetic masks, polishes or drug capsules. Allergic character of wheat gluten based products could limit the application for people suffering from celiac disease. Wheat gluten based films are having moisture, gas and solute barrier properties which are important in the areas of active coatings, active packaging, drug delivery systems and modified atmospheric packaging. Next type of plant protein based material is cottonseed protein films. Cottonseed oil extracted from cottonseed and oil extracted cake is utilized as livestock. This cake cannot be used for human consumption due to the presence of a toxic compound, namely gossypol. Cottonseed kernel contains 30 to 40% weight by weight of cottonseed proteins. Other components present in cottonseed are soluble carbohydrates, cellulose, lipids, minerals, phytates and polyphenolic pigments. Cottonseed proteins have a high content of ionizable amino acids and a low content of sulfur amino acids. These proteins are more soluble at a basic pH than at an acidic pH and have an isoelectric point of around 5. Next, we have to learn how cottonseed protein films are prepared. Initially, cottonseed films were prepared by soaking cottonseed kernels in hot water to prepare oilseed milk from which films were formed on the surface of the heated liquid, similarly to traditional U-bar films in East Asia. Owing to their poor mechanical properties, films made with this process may be used as edible items but not as packaging materials. Cottonseed protein based films are obtained by using a casting process directly from cottonseed flour. The first step involves solubilizing cottonseed flour proteins under appropriate conditions to minimize protein-protein interactions. Changes in the protein structure prevent remelting of macromolecules and reveal potentially interactive zones. The dispersions are then centrifuged to remove insoluble substances and the supernatants are homogenized. Glycerol is added as a plasticizer to the film forming solution. Drying allows solvent removal and the formation of three-dimensional protein networks through new inter and intramolecular linkages. Cottonseed flour films are having good mechanical resistance and insolubility in water. These properties are suitable for certain applications in non-food packaging, for example, compostable waste bags. They can also be used for agricultural packaging and mulching films to protect crops and fixate seeds and other applications where film color, porosity and biodegradability are beneficial. Other types of plant protein are also used for preparing edible films and coatings. Corn, soybean, wheat and cottonseed, peanut, rice, pea, pista and grain sorghum are the predominant types of plant proteins from which proteins with film forming ability are obtained. Now we will see about peanut protein. Peanut protein concentrates and isolates are commercially manufactured from defatted peanut flour. Peanut protein films have been prepared by two different methods. The first method involves the formation of peanut protein lipid films on the surface of heated peanut milk. This film production is similar to that of the traditional Asian yuba. The second method involves casting of peanut protein concentrate or peanut protein isolate solutions. Glycerin, sorbitol, polyethylene glycol or polypropylene glycol were added as plasticizers. Next type of plant protein is rice protein. Rice bran protein concentrates are prepared from commercially available unstabilized or heat stabilized rice bran by alkaline extraction and isoelectric precipitation. Byproducts of rice milling such as rice flour or broken rice kernels are used as rice protein by alkaline extraction. Enzymes can also be alternatively used for the treatment of rice flour or kernels to partially remove the starch component. Films from rice bran protein solutions have been prepared using glycerol as a plasticizer, adjusting the pH to 9.5, heated to 80 degrees Celsius, poured onto polyethylene plates and dried at 60 degrees Celsius. Let us see about pea protein. An industrial pea protein concentrate with 70.6% protein has been used to cast films with added plasticizers and dried at 60 degrees Celsius. Next is Pista Protein. The Pista Protein isolate is combined with hydroxypropyl methyl cellulose and polyethylene glycol as plasticizers to cast flames. The last plant protein is grain sorghum protein. The main protein fraction in the sorghum kernel is prolamine. It is also known as caffeine. Caffeine is having similar molecular weight, solubility, structure and amino acid composition like corn-zein. Laboratory extracted caffeine, which is having 89% of protein, have been dissolved in ethanol to produce films. Glycerin and polyethylene glycol have been added as plasticizers. The dried film are similar to those of films prepared from commercial corn-zein. Second part of this session is about animal protein based edible films and coatings. Milk proteins based edible films are tasteless, flavorless and more flexible. Transparent to translucent type edible films can be prepared by changing the formulation of milk protein. Main milk protein fractions are whey and caseins. Milk proteins possesses tremendous nutritional value and numerous functional properties which are required for the formation of edible films. Collagen, gelatin and egg white are the other animal based protein material used for developing edible coatings and films. Out of different type of animal protein, we will first discuss about casein. Milk is having 3% of casein. Commercial caseinates are created by adjusting acid coagulated casein to pH 6.7 using calcium or sodium hydroxide. Caseins are predominantly phosphoprotein that precipitate at pH 4.6 and at 20 degrees Celsius. Casein can easily form films from aqueous solutions and it widely used for various food applications. Let us look about whey protein. Whey proteins are soluble proteins present in milk serum after caseinate is coagulated during the cheese processing. Whey proteins represent around 20% of the total milk protein. Heat denaturation of the proteins can produce intact and insoluble whey protein films. Heat treatment is carried out in heating temperatures between 75 and 100 degrees Celsius using a protein concentration from 8 to 12%. Collagen and gelatin are the other important types of animal protein. Collagen is a constituent of skin, tendon and connective tissues of an animal. 30% of the total mass of the body is containing this fibrous protein. Collagen is the most commercially used successful edible protein film. The production of edible sausage casings in meat industry is carried out with the use of film forming collagen traditionally. Hydrolysis of collagen results in gelation. Gelatin forms clear, flexible, strong and oxidation impermeable films. Casting is to be carried out from aqueous solutions with added plasticizers. Industrial applications of gelatin include capsule coating and micro encapsulation. Edible coatings with gelatin reduce aggression, moisture and oil migration, or can carry antioxidant or antimicrobial agents. Last type of animal protein is myofibrillar proteins. Edible protein films have been formed from myofibrillar proteins from a variety of animal sources, but most of the work has been focused on fish as the main source of protein. Proteins obtained from mammal or fish sources must be washed and purified to obtain myofibrillar portion. Myofibrillar proteins have a higher tensile strength than other protein based films. Apart from the materials that we have discussed already, bioactive protein based coatings and films are also there. Let us discuss few points about that. Active compounds such as antioxidants and antimicrobials can be added into edible films and coatings. These bioactive films or coatings provide more inhibitory effects against spoilage and pathogenic bacteria and maintaining high concentration of the active compounds on food surfaces. Organic acids, essential oils, salts, lipids, spices and bacteriocins have been largely studied for their efficiency in controlling the growth of spoilage and pathogenic bacteria in foods. Let me summarize the points so far discussed in this given lecture. The first one is any type of material used for enrobing, that is coating or wrapping various food to extend shelf life of the product that may be eaten together with food with or without further removal is considered as an edible film or coating. Edible films can potentially extend the shelf life and improve the quality of the food systems by controlling mass transfer, moisture and oil diffusion, gas permeability and flavor and aroma losses. The next point is food components such as carbohydrates, proteins and lipids can be used for preparation of edible films. The next point is proteins are commonly used edible film and coating material. The fifth point is the plant proteins, namely extractable from corn, soybean, wheat, cottonseed and other crops are used as edible films and coating materials. Different types of plant protein based materials are widely used as: first one is gene film and coatings, second one is soy protein films, third one is wheat gluten films, fourth one is cottonseed protein films and the last one other protein films. And the last important point is milk, collagen, gelatin and egg white are animal based protein materials used for developing edible coatings and films.